Fibroin is the main protein in silk for moths and spiders. It's an extremely strong protein. It is a beta sheet structure. All these structural proteins, or all these proteins that we've been talking about up until now, have been helical proteins. Silk fibroin is an anti-parallel beta sheet protein. And if you remember, those anti-parallel sheets are stronger than parallel sheets. If you likewise, I'm going to go ahead and draw an anti-parallel sheet. That's going to look like this. And the reason is we've got parallel hydrogen bonds. It's got a bunch of small side chains, alanine and glycine, allow close packing of these sheets next to one another. The structure is stabilized by hydrogen bonding between those sheets and London dispersion interactions between those sheets. Here's kind of a cross section of what those different sheets are, and this is kind of highlighting those different small residues, those alanine and glycine residues. So if you remember, alanine is a methyl group. Here is that, and they're able to be in close proximity to one another. Glycine residues are kind of providing the space so that those alanine residues can be close together, because if there was something that was bulky right here or something like that, well, that would probably cause some repulsion of these adjacent sheets. So we've got these three different sheets that can pack very well together. Spider silk, used for webs, egg sacs, and wrapping their prey. It's an extremely strong material, stronger than steel, and can stretch a lot before it actually breaks. If you've ever walked through a spider web, you know that, yes, it can just seemingly stretch and go on and on and on and on and on. It's got a bunch of fibroin proteins and it's crystalline parts, so fibroin rich, as well as some rubber-like stretchy parts to it. Water-soluble globular proteins. This is kind of if you wanted to talk about the structure of proteins, this is the area that people are a little bit more interested in, because those other proteins like keratin and collagen, it's kind of predictable. You learn the structure, or you learn the amino acid sequence, and they follow a pattern. Now, globular proteins, they probably do follow a pattern, but it's a much more complicated pattern. This is to show the perspective of how different amino acids are going to pack and how they can actually look with respect to one another. So this right here is a representation of different proteins and how they would be displayed, basically the space that they would occupy if they were found in a specific configuration. So what you have is approximate dimensions if a single polypeptide chain would exist simply as a beta sheet. That would be 2,000 by 5 angstroms. If all of these amino acids in the same protein existed in an alpha helix, it would be 900 by 11 angstroms. Whereas a globular protein, you've got a 100 by 60 angstroms. So think this is a polypeptide chain that if you put it all into a beta conformation, it's going to be very long, but very short. An alpha helix, not quite as long, they can pack a little bit better, but they're going to take a bit more space. Whereas a globular protein, they're going to pack all of those same number of amino acids. So think about this as like a 500 amino acid protein. As a beta sheet, as an alpha helix, or as a native globular protein. Now, when you put those 500 amino acids in their native globular protein, man, they're going to pack very densely. It's going to be very efficient. It's 100 by 60 angstroms. So, when it comes to predicting, and this is the thing that I mentioned about patterns, when it comes to evaluating tertiary structure of proteins, one of the things that you have to understand is that there is a little bit of a pattern. So there are things known as motifs. They are also known as folds. And there are so motifs can be found as recurring structures in numerous proteins. Globular proteins are composed of different motifs folded together. So motifs are specific arrangements of several secondary structural elements. Motifs can be all alpha helices, they can be all beta sheets. They could also be a mixture of alpha helices followed by a beta sheet, or an alpha helix, beta sheet, beta sheet, beta sheet, alpha helix, or something like that. Now, these are examples of two different motifs. One of them is a beta alpha beta loop. And think about this right here as your N termini. You've got a beta sheet followed by an alpha helix right here. And then you continue that on to the C termini, and that's going to be your second beta sheet. So that's a beta alpha beta loop. This structure over here, letter B, that's known as a beta barrel protein. Beta barrel proteins are actually kind of the protein, or that's actually a structural motif that is common for GFP or green fluorescent protein. And in the center of it, that's where there's a fluorophore or a chromophore that actually produces that fluorescent light to it. Now, these repeated motifs contribute ultimately to the final fold of the protein. So when you think about knowing and understanding the secondary structure of a protein, it does have a contribution to the tertiary structure of a protein. So here's a beta alpha beta loop motif that's found right here. And that is found within alpha beta barrel protein. So it's kind of tough to see, but there's your beta barrel, and then here are all these alpha helices outside of that. So this motif does contribute. It's one of multiple motifs, multiple identical and similar, or identical motifs that contribute to that overall structure. It's a little bit tough to tell, but here is that central beta barrel. Okay, so this final fold is an alpha beta barrel.